Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending. I'm your moderator, Victoria Rees, editor of Drug Target Review. Today's speaker will be Dr. Jean Bernatchez, senior scientist and San Diego R&D group leader at Eurofins Discovery. Following his presentation, we'll be moving on to a live question and answer session where you can pose your questions to today's speaker. Please remember that you can submit your questions at any point during the webinar using the questions panel, which is situated in the menu on the right. But without any further hesitation, may I pass over to today's keynote speaker?
Thank you for the kind introduction. Welcome everyone, and thank you for choosing to attend this webinar on Eurofins Discovery's expanding E3scan platform for the identification of next-generation protein degraders. In today's presentation, I will be discussing targeted protein degradation as a new pharmacological modality, as well as presenting an overview of Eurofins Discovery's portfolio of assays to assist in the design of bifunctional protein degraders. I will show validation data for our currently available E3scan assays, as well as new data for novel E3 ligase assays we have recently developed. I will end with an overview of our compound screening capabilities and then take questions at the end of the presentation. Through our drug discovery experts, Eurofins Discovery can assist at the various stages of your drug discovery projects, providing services and products in the areas of synthetic chemistry, in vitro pharmacology, safety pharmacology, custom assays and products, in vivo services, and translational phenotypic services. While the scope of today's presentation will focus on our in vitro profiling assays, be sure to reach out to our customer service representatives if you're interested in any of our other offerings for your drug development projects. While major advances in traditional drug discovery have allowed for the inhibition of important disease-associated targets, 80% of human proteins have largely remained out of reach for therapeutic design due to their lack of a catalytic or regulatory site. The emerging field of targeted protein degradation, or TPD, has allowed for the targeting of proteins that were previously thought to be undruggable. The principle of TPD relies on the cellular machinery responsible for the natural cycle of protein turnover in the cell, described by the ubiquitination proteasome pathway. In this system, the E1 enzyme activates ubiquitin, which is then transferred to the E2 enzyme. Ubiquitin is then transferred to a target protein slated for degradation via the E3 enzyme. Polyubiquitination of the target protein marks it for degradation at the 26S proteasome. Ubiquitin is then recycled back into the pathway. In targeted protein degradation, a small molecule can be developed which hijacks an E3 ligase, directs it to polyubiquitinate a novel substrate protein through induced proximity, which then marks the novel target for degradation at the proteasome. Two popular strategies for TPD are shown on the right: bifunctional degraders and molecular glues. In the case of bifunctional degraders, one warhead specific to a substrate recognition domain of the E3 ligase is connected via a chemical linker to a warhead which can bind to a protein-protein interaction domain, allosteric site, or catalytic site of a target protein of interest. In the case of molecular glues, the small molecule can induce a conformational change in either the E3 ligase, target protein, or both proteins that reveals a surface where both proteins can bind together, facilitating ubiquitination of the target protein through induced proximity. There are several advantages of targeted protein degraders over traditional small molecule inhibitors. Targeted protein degraders can access previously undruggable targets, such as transcription factors, scaffold proteins, and other non-enzymatic proteins. they do not need to bind to an active site, they can be tissue selective, they possess sub-stoichiometric potency, and they have reduced off-target effects. However, only 1% of the over 600 E3 ligases in the human genome have been explored for TPD. As such, there is a large design space yet to be explored in this field for the development of novel degraders. The vast majority of bifunctional degraders, which have moved to various stages of clinical trials, utilize the E3 ligases cereblon and VHL for TPD, mostly due to existing ligands for these E3 ligases, which have been well characterized. However, these E3 ligases are ubiquitously expressed across tissues. Accumulating evidence has shown that some E3 ligases are expressed in a tissue-specific fashion, and that the nature of the E3 ligase recruited for TPD can be critical for the ubiquitination of a target protein via a degrader. As such, exploration of E3 ligases outside of cereblon and VHL can potentially unlock tissue-specific degraders, enhance potency of these therapeutics, and reduce toxic side effects. Eurofins Discovery has developed a panel of E3 ligase binding assays, known as E3scan, to assist with the discovery of novel E3 ligands for the design of targeted protein degraders. This panel can be used in parallel with our other binding assay platforms, such as KinomeScan, BromoScan, and BCL2scan, to identify ligands for target proteins of interest to design bifunctional degraders. Cell-based and phenotypic assays to evaluate the functional effects of designed protein degraders on the level of a target protein of interest can be performed using Eurofins Discovery's Sprinter, InCell, and Biomap platforms, and our synthetic chemistry services can assist in crafting the right protein degrader for your project. The E3scan platform builds on Eurofins Discovery's existing binding assay technology platforms, namely KinomeScan, BromoScan, and BCL2scan, and uses the same patented technology which has been successfully used by clients in a variety of drug discovery programs. In an E3scan binding assay, three key components are present: an E3 ligase tagged with DNA, a known E3 ligase ligand immobilized on a solid support, and a test compound or solvent control. The DNA-tagged E3 ligase of interest is incubated with a known ligand immobilized to the solid support. In the presence of a competitor test compound, the target protein is competed off the solid support. After washing of the solid support and elution of residual protein bound to the solid support, the amount of DNA-tagged protein is quantified via an ultra-sensitive qPCR readout. In the presence of a competitor test compound, the final qPCR readout is expected to have a low signal. In the presence of a solvent control, very little protein is competed off the solid support. After washing of the beads, elution of residual protein bound to the solid support, and qPCR quantification of the DNA-tagged protein, the assay signal is expected to remain high. A dose response of a competitor compound can be tested using this assay, and an accurate thermodynamic dissociation constant for the competitor can be extracted from the resulting data. E3 ligases in the E3scan platform are expressed in whole mammalian cell lysates as a fusion construct with the NF-kappa B DNA binding domain. An exogenous DNA probe is added to the E3 ligase containing lysate, which anneals to the NF-kappa B DNA binding domain. The DNA probe also contains the qPCR amplicon, which allows for detection of the protein of interest in the assay readout. We have validated our existing E3scan assays with small molecules from the scientific literature and have replicated published potency and rank order data for these compounds. The exquisite sensitivity of the qPCR readout of the assay system allows for the use of low picomolar amounts of protein in our assays. As such, our assays do not approach the tight binding limit, even for compounds with picomolar affinity, allowing for the measurement of a broad dynamic range of KDs during compound profiling. The E3scan platform is available in two formats. First, we offer ScanElect, where test compounds are screened at a single concentration and compound activity is provided as a % of an internal control. There is no curve fitting at this point, and a minimum 100 compounds is required per submission. The second format is KD-Elect, where compounds are screened in 11-point dose response in duplicate, and the data is provided as curves including KD binding affinities. We recently validated assays in the HECT NED4 subfamily of E3 ligases using capture ligands and control competitor peptides specific for the WW domains of these proteins and which contain the canonical PPXY recognition motif. Likewise, we recently validated assays in the BTB-Kelch subfamily of E3 ligases using capture ligands and control competitor peptides specific for the Kelch domains of these proteins. In a collaboration with the Gin Lab at the Icahn School of Medicine at Mount Sinai, New York, we profiled a bifunctional degrader and its prodrug, which were designed to bind to the BTB-Kelch subfamily E3 ligase KEAP1. We observed an almost 2-log difference in affinity between the prodrug and the active metabolite, highlighting the ability of the E3scan platform to identify affinity differences between analogs in a compound series. Eurofins Discovery now offers 20 E3 ligase binding assays spanning 12 E3 ligase targets. In addition, we have validated 6 additional targets in the BTB-Kelch and HECT NED4 E3 ligase subfamilies. These results demonstrate our capability to expand our E3scan assay offering to screen for selective ligands across E3 ligase subfamilies and to broaden the available degradable target space. Additional E3 ligases are currently being developed, and we are regularly expanding our assay offerings for this platform. There are major advantages in using the E3scan platform for TPD programs. Collected data are accurate, precise, and reproducible, and there is a high sensitivity and a broad dynamic range in KD measurements for competitor compounds. We have internal reference compounds that we have characterized for binding affinity, and all assays are run in parallel on a single platform, allowing for direct comparison of data between targets. This is the largest assay panel available on a single technology platform for E3 ligases, all without having issues of compound interference in the assay readout. Assays are performed in a high throughput 384-well format, allowing for library profiling. We also offer custom assay development on a case-by-case basis for your E3 ligase of interest. We offer rapid turnaround for compound screening of 5 days for weekly submissions and 20 days for library screens. As previously mentioned, Eurofins Discovery's biochemical platforms such as KinoScan, BromoScan, and BCL2scan can be used in parallel with E3scan during bifunctional degrader development projects. We offer 489 off-the-shelf kinase assays, 40 bromodomain assays and 5 BCL2 assays to screen for novel ligands for these targets. Furthermore, we also offer custom assay development services for clients on a case-by-case basis for assays not currently available in our catalog. As is the case for E3scan, we offer potency and selectivity screening for competitors, binding mode kinetic studies, and library screens for these additional platforms. Here is a flowchart for the general workflow for a custom assay development project. The initial stages, which involve cloning and synthesis of the immobilized affinity ligand and the control competitor, as well as the assay proof of concept and optimization, take generally 6 to 8 weeks, while compound screening, hit confirmation, and potency determination is performed in the 2 to 3 weeks following assay onboarding. In summary, I presented an overview of the technology platforms offered by Eurofins Discovery in the targeted protein degradation space, with an emphasis on the expanding E3scan biochemical binding assay platform. Profiling compounds on the E3scan platform can be coupled with compound screens on our KinomeScan, BromoScan, and BCL2scan platforms for the rational design and development of bifunctional protein degraders. With that, I would like to thank you all for attending, and I would be happy to take questions at this time.
Thank you, Jean, for your excellent presentation. We'll be moving on to our question and answer session shortly, where Jean will answer all of your questions. Don't forget that you can still submit any questions that you have for him using the questions panel, which is in the menu on the right. But before we get into the questions, we have a quick poll question for you. So if you could just answer the following: are you currently utilizing PROTACs in your drug discovery program? Simply select yes or no. Fantastic, thank you so much. So we can just see the results there that you've answered, with 60% saying yes and 40% saying no. Perfect. So let's move on to our questions. So Jean, the first question for you is, what is the turnaround time for custom assay development for the E3scan?
Hi, thank you very much, Victoria. The average turnaround is 6 to 8 weeks, as mentioned in the presentation, but that is broken down further into 2 separate sections. The initial cloning of the construct of interest and assay window determination step usually takes between 4 to 6 weeks, and then the further optimization of the assay and profiling of sample customer compounds to confirm in-house KD values that the customer may have would take another 2 to 4 weeks. After that takes place, then the assay is then transferred to production for screening.
Fantastic, thank you so much. So will you develop an assay that is not in your current menu and terms?
Yes, we regularly do this. We initially perform a feasibility search in the literature to see what does the state of the known literature on the specific target of interest. Are there published constructs, any structural data on the particular target or the domain of interest? Is there any qualitative or quantitative data pertaining to the affinity of a given capture ligand? all this information basically is taken into account in an initial feasibility study. If the project is feasible, we then proceed forward with a SOW, and then we then proceed to assay development.
Great, thank you, Jean. So the next question for you now, can the E3scan assay detect compounds that bind to another domain on the E3 ligase than the one that binds the capture ligand on the bead?
Yes, this is possible to do. You would basically only detect this if there was a possibility that the test compound was binding to an allosteric site and inducing a conformational change in the protein that caused the protein to dissociate from the capture ligand on the bead. It's more likely that you will be detecting in a given assay a compound that is directly competing with the site of binding for the capture ligand on the solid support. So if you are interested in looking at identifying compounds that bind to a distinct binding site from where the capture ligand for a given assay is bound, then it'd be probably best to develop a new assay, a custom assay that would be targeting that specific site, that site of interest.
Amazing, thank you. And just a quick reminder to our audience, there's still time to answer the questions, so please do send them in. So next question for you, Jean. For the E3scan, does the KD of the test compound, is the KD of the test compound dependent on the affinity of the capture ligand to its E3 target?
So for each of our assays, regardless of platform, E3scan included, but this is also the case for custom assay development and kinome scan or any other type of custom assay development we would engage in. We perform bait load gradients of the capture ligand on the solid support, and we assess the KD of the competitor at each of those individual concentrations of capture ligand on the solid support. And we establish an assay condition where the KD of the competitor no longer varies with the bait loading on the solid support. So for each of our assays, the KD of any competitor that is tested will not be dependent on the bait loading on the solid support.
Great, thank you. So do you have any plans to bring the E3scan platform from in vitro to a cellular format?
We already have existing assays that we have within our catalog, such as the Sprinter assays that I discussed earlier, that uses a CRISPR-Cas based system. I'd be happy to, after the webinar, direct you to our technical experts in that domain that can assist you with any interest you have in cell-based assay. But we are also in-house developing additional cell-based assays that re-implement this technology that we have.
Fantastic, thanks. Another similar question, do you have cell-based assays for degradation? There might be some crossover with the last question there.
Yes, yes. As I presented in the presentation, Sprinter would be the one, yeah.
Fantastic, well, thank you so much. Please do join me in thanking Jean for his time today. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. Please do take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. And if you could complete it when you can, we'd greatly appreciate it. So thank you so much, Jean, for your time today. It was great to speak with you.
Thank you very much, Victoria, and thank you very much, everyone, for attending.
And on behalf of Drug Target Review and Eurofins Discovery, I'd also like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.